All right. Well, good morning or good evening or good afternoon, everybody, depending where you are in the world, which it looks like we have quite a global representative group today, which is super exciting. I always like at the start of these webinars, seeing where everybody is joining us from. So it's been fun for me, at least, to see these bubbles show up on the map, though I recognize it's probably a little tricky to drop the pin on it, but at least I get a general sense of where everybody is. So thank you to those of you that participated in this poll this morning. Well, welcome to one of our first advanced webinars of the calendar year for FlowJo Software. My name is Veronica Obregon Perko, and I am a senior scientific advisor for informatics with BD Biosciences. So not only offering consultation and education for FlowJo Software, but some of our other platforms that support flow cytometry experiments and single cell analysis. And in today's webinar, as you saw in the description, and I'm gonna go ahead and close this screen, but I'll leave the poll open for those of you that are still kind of working on dropping your location there. But I'm gonna navigate to my PowerPoint now. I can get out of this screen. There we go. All right, so let me go ahead and navigate to my slide deck, so we can remind ourselves on why we're all here today, which is basically to talk about advanced gating tools in FlowJo Software version 10. So we have plenty of videos on our website, which I'll show you where to find those at the end of today's session. But there's plenty of information on how to, initiate a basic analysis, basic identification of populations, all the way towards making a report. And then we also have a lot of videos on the far end of the learning spectrum in terms of level of advancement for the tools in FlowJo software. And that's going to be more like unsupervised analyses like dimensionality reduction and clustering. But what I'd like to spend time on today is everything that kind of sits in between, right? Are there opportunities to kind of bump up the efficiency of a basic analysis, maybe shortcuts to find different populations? And also, do we have an opportunity to maybe automate some parts of the analysis? So we're not always doing so much manual work, and when is it applicable and when can you use it? And what are the requirements and assumptions? So that's what I'm hoping to spend our time on today. So a couple of housekeeping things. So I'm going to kind of divide our time today amongst PowerPoint presentation and then doing a demo in the software. So I'm hoping to wrap up around the top of the hour. But if you need to leave early or step away from the computer at any point, don't worry, this session is being recorded, and the recording typically gets posted on our learning section of our website, usually within about a week at the most, but it's typically within a couple of days. So you can always go rewatch this content at any time and revisit what we discussed. This is fairly informal. If at any point you have any questions, don't hesitate to put them in the chat. You don't have an ability to unmute and ask your question, but you can type out your question in the chat and I'll do my best to answer it in the moment, so we don't have to hold all questions till the end. If you think of a question after today's session, I've included my email address here. Feel free to email me directly with any questions, or if you need a quick demo or recap of anything that was discussed in today's webinar. Okay, so that being said, let's go ahead and get going. So let's go ahead and start with another poll. So we already did one that I thought was rather fun, which was just to kind of see where everybody is in the world today. But let's do another question. And really, in addition to my curiosity of where everybody is joining us from, I'd really like to know what's going to be your learning objectives for today. So for those of you that joined a little late and didn't see the join instructions before, I'm putting them here for you again. So two ways to join the poll I'll be using throughout the webinar. You can go on your web browser on your phone or laptop, go to menti.com and enter the code. Or if you'd rather just be on your phone or you have some kind of a nifty tablet with you, you can also scan the QR code and that will take you directly to the poll as well. Just go ahead and leave it open and that way you can just return to it when we pull up the questions again. Okay, so I'll give people just a few seconds to do either of these and then I'll proceed to the question. Okay. Great. So let me go on to the question now, which was really just, I just want to get a sense of what topic you're most excited about, right? Are you here today after you saw the session, the title of today's session? Did you, decide to join because you want to learn how to speed up your analysis? Do you have some complex populations that you want to learn how to capture, perhaps first getting started or just learning how to optimize that? Or are you looking to just try to automate parts of your analysis, right? Let computational tools kind of help you along, even if they don't do the whole thing, but maybe they do parts of it. So I'll give everybody a second just to kind of, tell me what their main expectations are from today's session. But the good news is we will touch on all of these, but I just want to get a sense of what people are most excited about. And it looks like it's a fairly even spread so far. Lots of excitement about the automation, which I'm really glad I included that. I was scared I wouldn't have time, but I'll be sure to make time now seeing these responses. Okay, I'll give people just one more second. All right. And again, I'll leave this open in the background. You can still participate if you're working on your answers, but I'm going to go ahead and migrate now to the PowerPoint deck so we can get started. All right, so let's go back into our deck. I'll turn on a laser pointer for us. All right, so what's wrong with basic manual gating, right? In short, nothing really, right? It has a lot of advantages over something like letting clustering tools, which are automatic population identification tools, doing that work for us, right? So what are the advantages of going with manual gates to begin with, right? So if you're arguing with a bioinformatician who says that you should just use a clustering tool to do your analysis. Well, some of the advantages of basic manual gating is that the populations get user defined, right? So it's a very straightforward approach to identify some specific phenotype that's based on the literature or based on your knowledge of cell types that you study in your lab. So there's no question really of what the phenotype of a cell is because the phenotype has been user defined. So you can very clearly explain what that cell type is expressing. You can align phenotypes with established biology. It's easy to talk to people in the field about what a CD8 T cell is if you both manually gated for it in the same way versus if you both found some kind of rare CD8 T cell with a clustering approach that now you have to kind of compare approaches and really deeply look at those phenotypes to see if you both are in fact studying the same cell type. And at the end of the day, we get gates, right? The population definitions can be modified by adjusting gates, right? If I decide I want the cell to be, or I want the gate to capture a CD8 low versus a CD8 high, right? It's very easy to just move those gates and change those definitions. But with most clustering tools, we don't get that opportunity, right? You get the populations back, but it's just a population, right? There's no way to supervise the clustering tool and say, oh, can you actually make the cutoff at CD8 here and the cutoff for CD95 there? So it's not as straightforward to make those tuning adjustments, right? But when we have gates that we ourselves have drawn, it's very easy to just move them to the left, move them to the right. Now, that being said, there's some considerations, right? We have to still establish those gates and we have to nudge gates between samples, right? So at the end of the day, if I have sample one and I'm trying to capture the population here on the right, it's not as easy as just copying and pasting the gate onto sample two, because you can see that when I've done that, I've excluded part of that population, right? There's a shift in not only the location, but the size of that population. And so I'm going to spend a lot of time as a cytometrist nudging gates between samples to account for sample to sample variability. There's endless biaxial combinations, right? So this is just one biaxial combination, right? So one axis, two axis, that's biaxial. So you're only ever looking at two parameters at a time. And if you have a rather large panel and there's a lot of activation markers and things like that, or intracellular cytokine staining, you're going to be doing a lot of gating on plots that look like this, right? A lot of biaxial combinations, marker A versus B, A versus C, B versus C. And depending how many markers you have in your panel and what these cells can be co-expressing, that's going to be a lot of gates to draw. And even after you have identified possibly shortcuts and things to help you, establish these gates, it's still going to be a lot of work if you have a really large study. Okay, so at the end of the day, manual gating, when confined to just the basic approach, is great because it directly gets you to a phenotype of interest, but it can be an investment of your time to basically optimize the coordinates at that gate between your samples or between studies. So the good news is there's some things we can do to kind of boost the productivity a little bit, right? And try to lift that burden of really just the time-consuming effort of moving gates either within a hierarchy or between hierarchies, between samples. So these are kind of the three categories we'll be talking about today. So we're going to spend a little bit of time talking about clone gates, which give us an opportunity to synchronize gates within a sample. We'll also talk about combinatorial gates that we divide into Boolean and merged gates, but they're a means to the same end, which is basically to use single gates to define complex populations, right? So if I already have single gates established, I can kind of use logic to combine those and identify populations that are expressing markers and various combinations of those established gates. So not having to start from scratch, but just kind of leveraging what you already have. And then we'll talk about automating our analysis a little bit. Is there an opportunity for populations in my data to be identified automatically, but maybe not give so much freedom as you would give to an unsupervised clustering tool like FlowSum or Phenograph, but instead you as a scientist teach that tool how to find populations in your data based on what you know and what you want the tool to retrieve for you. So something we refer to as supervised auto-gating. Okay, so we'll spend a little bit of time on that as well. So clone gates, right? What are clone gates? Speaking honestly, when I first joined the FlowJo team, I was exposed to, FlowJo Software 10, and that's what I've kind of spent my flow cytometrist career in. And I kept hearing people talk about clone gates, and I had no idea what they were because it turns out they were a feature that was in version 9, and I believe it did not get added to FlowJo version 10, or at least earlier versions of FlowJo version 10. And so I had no idea what they were, but apparently there was a big outcry from the flow cytometry community to bring clone gates back. And I think you'll see why, because they serve a very specific purpose. And I'm really glad that we have it back, because now I can really see using them a lot in analyses that I help users with. So what is a clone gate, right? And why were people so upset when they disappeared for a short amount of time? So clone gates give you an opportunity to synchronize gates within a single sample's gating hierarchy. Now, to be clear, this is going to be distinct from synchronized to group. Okay, so when you create a group in a FlowJo workspace, and I'll try to remember to show this in the demo, when you create the group, there's going to be a little checkbox at the bottom, and it'll say, synchronized to group. And what that does is when you turn that on, it's typically off by default. When you turn it on, if you make a change in your hierarchy to one of your samples, it will apply to all the samples, right? Which might be what you want if these are like replicates, for instance. But in this case with clone gates, we're not synchronizing to the whole group. We're just synchronizing 2 gates within one sample, right? So what would be the use case here, right? So for instance, let's say I've got this hierarchy here, I've got this sample, and I'm looking at interferon gamma expression, and it's the same interferon gamma marker and antibody and everything, but I'm gating it on CD4 cells and on CD8 cells, right? So 2 separate kind of lineages there, but all still in the same sample. And so I would presume that that gate needs to be in the same place for both of those populations, right? And maybe I've established this cutoff on a fluorescence -1 control or something like that. So what I can do is select those 2 interferon gamma gates within the same sample, right click, turn them into clone gates, and you'll see that now you have this kind of like clone 3-diamond icon preceding the gate name. And what will happen is that while I'm in my analysis, if I nudge this gate on the left, the gate on the right will move exactly the same, right? So now these gates are in complete synchrony. So you can see what a shortcut that would be if you had, let's say I had this CD4 population, but I had different memory subsets, right? Or different types of activation statuses and things like that. And later I decide to make an adjustment to one of my interferon gamma gates, right? Without clone gates, I'll have to go into each of those separately and make those little adjustments. But if I clone them all, I only have to adjust one and the others will inherit the adjustments. And so again, caveat here is we would expect the gate needs to be in the same place, right? If these populations, for whatever reason, have completely different levels of spread, like that negative peak looks much wider, and the CD8 population, for whatever reason, right, then clone gates may not be what you need. But if you have something like this, they're going to be a nice time saver for making gate adjustments. All right, so so that's clone gates. And a moment ago, I think I saw somebody raise their hand, but just a reminder that I can't unmute you for you to ask a question. So if you do have any questions, just be sure to put it in the chat and I'll keep an eye on it. I don't see any questions yet, but make sure you post them there. Next slide. All right. So now let's talk about Boolean gates, right? So Boolean gates give us an opportunity to simplify the gating of complex population using inclusion or exclusion logic from existing gates. Right? Oh, I think I see something in the chat. Oh, yes, there's a question in the chat that says, can you unclone a gate that you cloned? Yes, absolutely. And so if you want to just do it temporarily while you make your gate adjustments and then go back and turn it off, it's a simple right-click function. So it's not permanent. It is reversible and reapplicable as well. Let's see, there was another question about, let me scroll up a bit. Never knew this was a feature. I've been using it for 20 years. Yeah, I'm right there with you, William. I haven't been using it quite that long, but I've been using it for a long time and was also surprised when someone was like, bring back clone gates. And I first had to ask what it even was. So I'm glad it's back. Let's see, there was another question. Do we just do that sample or sample by sample? Yes, Vaishnavi, that's a good question. So let me go back to my clone gate slide here. So what I would advise is that you're gonna want to clone gates within a sample. And I don't even think the workspace will let you clone gates between samples. It has to be within the sample. And really that's because we would expect gate placement to be the same within a sample, right? If we start talking about between samples, now we have sample to sample variability, which we have other tools to deal with that I'll talk about in a moment. But this would just be on a per sample basis. So then here in this screenshot, if I had another sample below there, I'd pick those interferon gamma gates in that sample and clone those. Okay. Now let me go see if there was another question. Oh, there's a few coming in. Okay, let me make this a little bigger so I can see. Okay. We just do that. So I answered that one. Okay, another question from Anna is, if we then apply gates to the group, will it change all clone gates or will it apply just to that subpop? Let me read that one more time. If we then apply gates to the group, will it change all? Ah, I see what you're saying. Okay, so the workflow, Anna, that I would recommend is to probably group apply your gates first, right? So finish your hierarchy, group apply the gates. And then when you start getting to the point of wanting to nudge your gates around, like adjust them sample by sample, at that point, I would turn those interferon gamma gates into clone gates at that point. And then there was a question about how do you clone the gate. Gabriella, we'll go through that in the demo, but it's basically a simple right click. So if you multi-select the gates you want to clone, you right click and there'll be an option to clone gates. And you'll know that the gate's been cloned because there'll be these little 3 diamond symbols on there. I think I answered both of those questions at the same time. Somebody asked, Ariella asked about the recording. I'm not 100% sure, Ariella, if you get the recording by email. I want to say yes if you pre-registered, but if that doesn't come through, you can retrieve the recording from our website in the Learn section. But I think maybe you do get a follow-up email with a link to the recording, but if not, it's easily found on our website. Okay, it looks like last question here. If we are basing gate on FMO, why wouldn't the gates stay the same between samples? That's a good question, Kelly, and that really, that could just be a topic on its own webinar, really, for like basics of data analysis, but the reality is that when we have various samples in our experiment that come from different human donors or come from different non-human primate donors and things like that, Sample to sample to variability is normal, right? We will sometimes see population shift to the left or the right a little bit, or you might have a donor that has way more CD8 cells, and the abundance of that population has now changed the shape of the negative population when you're gating for some kind of activation marker. So it's pretty well established that sample to sample variability is there. It can be minor, but it can be minor enough that, or it can be major enough, I should say, that we can't easily just copy and paste gates from a single FMO unless you have an FMO for every single donor in your experiment, which is usually not the case, right? We don't always have the resources for that. So while we would expect them to be generally in the similar area between samples, it's not as simple as just picking a cutoff from an FMO and saying, oh, it's at 10 to the third in that FMO. So in all my samples, I'm going to put it at 10 to the third. Because you'll see that if you stimulate your cells, if some cells are cryopreserved and some aren't, some donors have more of a cell type than others, those populations will shift. And so gate nudging is very well known in the cytometry community, right? When we're working with things like cell lines or even some strains of mice, we might not see it as strongly as we do in models that have a lot more genetic variability. But I'm happy to, if you send me an email, I can send you, papers on this topic and things like that. But how I wish it was as easy as just copying and pasting gates between samples and we didn't have to move them at all. Oh, I think there might be something in the Q&A. Oh, okay, yeah, there's also a Q&A box. I'll try to keep an eye on both, but the chat might be a little bit easier for me to look at. There's a question in the Q&A that says, if you clone a gate, can you adjust it after? Absolutely, and that's the whole idea, right? So if you do the clone gate and then you make an adjustment, that adjustment will be inherited in the populations that have that clone gate. So yeah, you can absolutely move that gate in either direction after you clone it. Okay, I'm hoping I got everybody's questions. A lot came through at the same time. So glad to see so much interest around clone gates. Okay, let me check. I might have missed one more question. Oh, answer live. Done. Okay. Okay. All right, well, let's go on then. And go to my next slide. Right, so we were about to talk about Boolean gates, right, which are basically a way to capture complex populations if you already have existing gates, right? So those kind of go into two different classes. You might have noticed them before in your FlowJo workspace ribbon at the top. There's a Boolean band that has all of our Boolean gate options, right? And I've divided them into kind of two categories here. So the first one is going to be inclusion or exclusion logic to basically capture populations that already exist right in the workspace, but capture them in different combinations. So let's say with the use cases, right? So we have Boolean, or, and not gates. And my favorite thing about the UI of the Boolean gates is that the little icons kind of give you a hint of what's going to happen when you pick that function, right? So it's a nice little shortcut if you feel like you can't get the logic straight in your head. So one of my favorite ones to use is the Boolean not gate, right? So what would be a use case for that? So let's say I have a panel and I'm staining some kind of tissue suspension. And a good amount of those cells are going to probably be lymphocytes, but I don't want the lymphocytes. I'm actually interested in, maybe like the epithelial cells, platelets, things like that. And so I want all of these cells that aren't lymphocytes, but that's going to be a really awkward gate to draw, right? To have to capture everything that's not a lymphocyte, right? I can identify the lymphocytes very easily, but those aren't the cells I'm going to use in my downstream analysis. So what you could do is gate the lymphocytes, turn that gate into a Boolean not gate, which basically means I'm drawing the gate, but I want everything that's not in the lymphocyte gate. And the result is what you see on the subsequent plot, right? Now I've captured this kind of awkwardly shaped population and have completely excluded the lymphocytes that were inside that gate. So a quick way to capture something that you want, but maybe it's easier to gate the other population that you don't want. Right? So the other situation you could see using this in is for those of you that prefer to draw time gates manually, where you're doing quality control on your sample. Maybe you have time on the x-axis, a parameter on the y-axis, and you're trying to remove these periods of instability, you could end up drawing kind of like a W-shaped gate, which might be awkward, but instead you could draw a gate on the period of instability, say not, and then take everything that looks nice and stable, right? So that's probably my favorite use case for a not Boolean gate. And so that's basically selecting the gate, going up into the ribbon and picking one of the Boolean options. In this case, it's a not option. Now I'll come back to the and or because I feel like it's easier to explain the combinatorial option first and then explain why we have the or and options. But you can see from the icons what they're doing, right? Basically, you're either taking two populations and saying if it's either this or that, whether it's single, double, or double negative, double positive, single positive, you want all of it lassoed together, or something that has to be co-expressing, right? Those two markers, or it fits the both criteria. A combinatorial Boolean gives you an opportunity to basically get everything you see on the right, right? It's going to quickly give you all the positive, negative combinations so that you don't have to capture them manually, right? So you'll hit that create combination gate. And here's an example of what it's doing, right? One of the most popular use cases for this is for people that are looking at different T-cell subsets or T-cell types based on intracellular cytokine staining, right? So here in this example, I've individually gated for, let's say, interferon gamma, phosphoric, and perforin. Ultimately, I want to find the cells that are double positive for interferon gamma and perforin, and maybe a bunch of other different combinations as well. So I select those two populations, go to the create combination gates, and now I have all of those populations defined for me automatically. And so this is a plot I made in the layout editor, and I overlaid all these populations just to show you the phenotypes, right? So you can see that it found double positive, double negative, and single positive for each of the cytokines. Now here you're getting all the combinations. Now let's say you only wanted the purple population, right? This is where the individual Booleans come in handy. So if later in my analysis I decide, oh, I wish I had done a quadrant gate and just done interferon gamma double positive for perforin, I could just go select those two populations, make a Boolean and gate, right? And now I'll get that population. Okay, so you'll get them all automatically if you do the combo option, but if there's just one specific phenotype you want, you can make use of some of these others, and you don't have to clog up your workspace with populations that you're not interested in. Okay. So what are some considerations for Boolean gates? Boolean gates do take more processing power than a typical manual rectangle gate or something like that. Now there is an option in your preferences, and I'll try to remember to show that, where you can cache Boolean gates, which tries to keep them in the FlowJo memory, and that way when you relaunch your workspace or make calculation changes, you don't have to wait for it to load all of the new frequencies. But just keep in mind that I wouldn't go crazy with Boolean gates and make all these combinations of cytokines if they're not going to be used in my downstream analysis, because you're adding kind of a lot of computational burden to the workspace. So I would just say use what you need and kind of leave behind what you don't. And you're not going to get any resultant gates. So for instance, if I open up this double positive population for interferon gamma and perforin, I'm not going to see like a quadrant plot, right? Because these were not established with quadrant gates. These were established with Boolean logic, okay? So if you do want to make adjustments, you have to go back to the original populations that derived those Boolean populations, okay? But on the next slide, we're going to talk about an alternative that gives you a quadrant gate display if you're very visual and you want to see that distribution all in one plot, right? I got this visualization in the layout editor, but it was not given to me automatically. All right, let's see what we have in the chat here. Okay, and William says, in version 9, you can make Boolean gates of separate subsets, such as to make a Boolean gate of CD4, CD8, double negative, which all express an NK marker. In 10, it adds all of the Boolean gates under one of the populations, okay, such as all under CD4. When you open the population in the workspace, you do see what you would expect with all the populations, but then you put that stat derived from this into the table editor. It now only shows the events which are under the CD4, not the combination of all three. Let's see, William, I'll have to maybe revisit this question because I'm not sure I understand what the question is. I believe you can do Boolean populations for subpopulations, but perhaps I'm not understanding the question very well. Adds all the Boolean gates under one population, such as under the CD4. It says in version 9, you were able to make a Boolean gate of CD4, which all express an NK marker. Yeah, again, it was my understanding you could do them under individual populations, but I will get back to you on that. And if I have time at the end of the webinar, I'll try to revisit the question. See, there's another question that comes through that says, can we get statistics from the Boolean gates? Yeah, absolutely. So I cut it off here in the screenshot, but you will get the frequencies and any other stats you're interested in, like MFI or count, any of the traditional statistics we typically add to the workspace. Yeah, you can treat that as any population. The only difference that I just want to be clear about is that you're not going to see a display like what's on the right, like a typical quadrant gate, right? So you get the populations, but you can't adjust these with typical quadrant gating, right? You just go back to the single populations and make any adjustments to those gates, and those will adjust the resultant populations. Let me close the chat, and I think I saw also something in the Q&A. Let's see. Where do you enable the cache? I will show that in the demo, but it is basically in the preferences, which is that heart-shaped symbol at the top right of the FlowJo workspace. I believe it's either in the gate section or the performance, but we'll look through that when we get to the demo. Okay, so I think I covered everything I wanted to here. So again, there's individual Boolean gates, and then there's the combination, which basically just kind of does everything. You get the resultant population, but no quadrant gates to modify, right? You're just modifying the original populations that derive those. Now, what's another alternative to Boolean gates? We now have, and I believe this is new to version 10.9. It might be 10.10, but definitely if you're on the latest version of FlowJo, you'll be able to see these merged gate options. Okay, they're relatively new. So merged gates are kind of an alternative to Boolean, where they allow us to create combo populations, but we get kind of a resultant quadrant gate, right? So this was actually developed by one of the members of our product innovation team, where he would have customers that maybe had this really extensive analysis that included bisector gates, so they preferred to do all of their let's say cytokine gating on a single parameter view, so in a histogram, and they have something like IL-6 negative positive, IL-12 negative positive, and so they have all this work they've already done, but have decided that they need to change gears, and now they need to see that as a distribution on a quad plot, right? We need to know which cells are double positive, single positive, and double negative, right? Do I have to start from scratch and go all the way back to my parent and reinitiate these quadrant gates, or is there something else that can be done? So that's really what was kind of the motivation behind this tool, which was to have users not have to start over if they decide they want to change the way that the data is displayed or gated. So we kind of went through the use case already, right? So the use case would be you have a series of bisector gates, and these need to be bisector gates, right? A bisector gate means you divide all the data on the plot to the left and the right. And the reason it needs to be a bisector gate instead of just like a manually drawn range gate is because we're going to have to establish the quadrant, right? So if you do a range gate, usually something gets left behind, and we can't leave events behind, right? Everything has to be accounted for in the subsequent quad gate. So here you'd have two sets of bisector gates, and then you can select those sets and say that you want a merged quad gate or a merged curly quad or a merged rectangle, right? So if you're not even interested in the other combinations and you only want the double positive or something like that, right, you could lower the computational burden of quad gates and just stick with the rectangle gate, right, and get that combination. So again, unlike a Boolean, here we are getting a resultant quad gate, we are getting resulted gates that we can modify. There is also a cool auto merge quad function. So in this case, you don't even need to have established bisector gates. If you have the CD14 monocytes in this example, I could select the CD14 and say, I want you to automatically make me two sets of bisector gates as well as a resultant quad gate, right? So here you just pick the two parameters. You say how you want the threshold established, right? Valley point, I think, is going to be the most applicable most of the time, which is just when you start to see the dip between the two peaks. And then if you want curly versus regular quad, you just hit the checkbox. And so with this tool, you'll get the bisector gates and the quadrant gate. Okay. So again, another consideration, we don't want to overuse these kinds of tools, right? We'll use it when it's needed. Quad gates and curly quad gates do take some processing power. So the more of them you have in your workspace, especially if you have a lot of samples or a lot of events, the calculation time when you make any kinds of adjustments is going to be a little longer than you would see just with a standard rectangle gate. Okay, so again, use it if you need it, but I wouldn't just throw a bunch out there, you know, just for the sake of fun. All right, let me check the chat again. Okay, let's see. William added a little bit more to his question. Let me see if I can address that in the demo. It sounds like I'm trying to make an NK expressing T cell population by adding separately gated CD4, NK, CDNK, and TMI populations together. Oh, I see. I think I'm starting to get closer to understanding the question. I see. Okay. Okay. Yeah, let me see if I can address this in the demo, William. And if I don't get to it, I will try it on my own after the webinar offline and see if I have a workflow that can address this. It was an interesting question. Yeah. Okay. I think we went through all the merged gates. Okay, let's see how we're doing on time. Okay, great. So I'll start to wrap up the slides here basically by, you know, introducing an opportunity for supervised auto gating, right? So I think we're all, you know, now being in an advanced webinar, are all pretty familiar with what manual gating is, right? It's strictly user-defined populations, and we can make use of tools like rectangles, polygons, and even some of like the Boolean and clone options we talked about just to help, you know, speed up gate placement a little bit. And then all the way on the other spectrum of population identification, there's things like clustering tools, which we have a lot of high parameter webinars or high dimensional analysis webinars on our website that cover these way more comprehensively. But just to say that clustering tools are largely unsupervised, right? You don't have an opportunity to teach the clustering tool what a CD8 cell should look like, what a CTL should look like. You're kind of just letting it use the visual separation of populations in the graph space, in the high dimensional space, and kind of identify populations based on their profiles and expression patterns of the markers in your panel. But you don't get any gates, right? You can't make adjustments and you can't supervise. But now there's kind of a class of tools that sit somewhere in the middle, which is basically like, I want computational tools to help me find my populations, but I want to help guide it. I want to teach the tool where I think the cutoff should be for a population. And I want to only find populations relevant to me. I don't want just 100 populations returned back, but I'm only interested in CD8 populations or something like that. And so this is where these two tools fall into that category, right? So we've got two of these available as plugins to use in FlowJo software. One is called BD Elastigate, and the other is called Easy Daffy. So I will pause here and say that we have an entire supervised auto gating webinar on the FlowJo website. You can also find them on YouTube. And that's kind of a 1-hour deep dive on these tools. So I'll be going through them a little quickly today just for the sake of time, but just know that if you want to dive into those longer, those recordings are available. So quickly, I'll go through Elastigate first. I think this tool is really just trying to mimic the visual process of gate nudging, right? So mimicking the work that a cytometrist would do during their analysis. So the use case would be you have a well-defined hierarchy, right? A group of cells that you find with some reliability. You've invested a lot of time into finding those kinds of cells in one or more samples. And you want to use those samples to basically train the tool to find all those types of populations in new samples that are going to have similar biology, right? And we'll come back to that caveat in a second. But the way the tool works is it basically creates images of your plots. So let's say I have the training sample, which is the sample I'm very confident about my gating on, and then the target sample, which is the new data. And so we basically transform these plots into images, right? Pictures. Then a technique gets used called image registration to transform the training image into the target image, right? So how do I have to shift these populations so that they match the target populations? And then that gets basically turned into, kind of like these vertices. And then the vertices get applied to the target sample's gates, right, which were inherited from the training sample, but now we apply the transformation to accurately capture those populations, right? And notice how these two plots look similar, right? But back to the question we had earlier in the webinar, which was, why would you expect sample to sample variability? It just happens, right? That's just biology, right? These two plots look very similar, but I still have this, upper population that has shifted a bit. And I have this population on the bottom left that's a little bit wider than on the right, right? And that could be because they were processed on different days, or they're just different donors and they have different scatter properties. And so this is going to be kind of the gold standard tool for addressing sample-to-sample variability in a computational manner, right? So letting a computer help you move those gates around. And the best part is you still get the gates. So if you don't agree with the gate placement, you can still make adjustments after the fact. Now, considerations for the tool, it doesn't support Boolean gates. Okay, so it needs gates to adjust. Remember, Boolean gates, even though we say they're gates, they just give you the resultant populations, right? We don't get a gate to directly modify. So you can't use it on those Boolean populations. It's going to be less effective on kind of sparse or rare populations. So if you have like a tetramer specific T cell, that's like 0.01% of the data or something like that, it may not do as great a job, right? Because we need more events on the plot, right, to render the image. I say missing populations, but what I mean by that is kind of going back to this thing about similar biology. Okay, so if you train Elastigate on an unstimulated PBMC sample, and you try to apply it to a stimulated PBMC sample, right? Some populations are going to appear and disappear relative to the unstimulated, right? When we stimulate cells, they start upregulating things. Right now we have activated T cells that weren't there before. And because we're turning everything into an image, and the algorithm kind of assumes, well, I see a big population here, and now it's down here, but it must be the same one. That might not always be the case, right? It could just be that the population wasn't there before, or it's there now, but it's not the same as what was used in the training sample, right? So it's going to be good to train on similar biology as much as possible. We do have a kind of a setting in the algorithm when you run it to help address these things. But of course, you want to set the tool up for success and try to train and apply on samples that are similar. Okay. The other case would be FMOs, right? In FMOs, fluorescence -1 controls, populations are intentionally missing, right? Because we use those as a negative control to establish gating. So if you have a CD4 FMO, right, you won't have any CD4 cells. So Elastigate might get confused when it can't find them, and it might apply the gate to a CD4 negative population. Okay, so those are the kinds of examples that I'm trying to convey here. But overall, really fast. I don't see the harm in trying it in your data because, again, you can always undo the changes and you can always make adjustments to the gates. Now, EasyDAFI is a pretty cool plugin because it's almost like you're building your own custom clustering tool, right? So with Elastigate, we're mimicking the cytometrist workflow, right, of visual gate nudging. With EasyDAFI, we're basically saying, I don't care about the gates, I just want the populations. And even if that means you have to use markers I didn't think about or I didn't include in my hierarchy, that's fine. But I want cells that match this phenotype that I've gated for, right? So let's take a look at how that algorithm works. So here's, let's say, your data represented in the high dimensional space. And when we say that, each dimension is one marker in your panel. So if you have 10 markers, you've got 10 dimensions. So you've got that data. The tool will run a clustering in the background. So it will automatically find populations, right? They're all represented with these different colors. So here it found 1, 2, 3, 4, 5, 6 different populations. Now I come in and do a manual gate, and this is my manual gate, this box. It includes population pink and population blue. And so now I'm teaching EasyDAFI, these two clusters are basically the same population, and I want you to treat them as the same. And so now they've turned red, right? Because this is my new Veronica created easy Daffy defined cluster. Or I should say Veronica defined cluster that easy Daffy will try to find in subsequent data sets, right? So now I've taught it how to find these cell types, and it knows Veronica's red cell type can be defined by these blue and pink, cluster populations. So again, use case would be, right, you want the populations. You don't care if you have the gates because you're not going to get any resulting gates, but you're okay with letting a computer or an algorithm try to find those populations for you. And maybe even some novel ones you hadn't really thought about, right? Maybe you were focused on CTLs only based on their perforin and interferon gamma expression, but Easy Daffy can find those cell types also based on their CD38 expression as well. Okay, so it will also consider markers that maybe you didn't directly include in the manual gating. Okay, so a little bit more advanced, but gives you room for computational discovery while still supervising that manual gating. Okay, something in the chat here. What is the min and max number of parameters needed for Easy Daffy to ideally function? Oh, that's a good question, Savannah. I feel like I go over that in the supervised auto gating webinar. Oh, I'm trying to remember. I do think there is a minimum. I mean, I think as long as you have a handful, that's fine. Now, that being said, if I have time in the demo, I'll show this. When you run Easy Daffy, it will ask you if you want to use all the parameters or have it only use the ones that show kind of like a discriminatory power, and that kind of lowers the computation time of the tool and also makes it more effective. So even if you had a large parameter data set, Easy Daffy might not leverage all those parameters to identify these cell types. It might find that only 4 of them are necessary to find those clusters that were interesting to you. So that is a toggle in there. I mean, in terms of the minimum, I really can't remember that offhand, but I think if you have at least a few, it should be fine, but I'd have to get back to you on that one. Okay. So I believe that's it for the slides there. So I did put a quick little quiz here. Let's see if we can get through it in a few minutes, and then I'll try my best to go through the demo. We might go a little bit past 11 if you guys can hang with me till like 11 my time. So maybe we'll say 15 or so past the hour. If you guys can hang on there, you can see the demo. Otherwise, if you need to drop off, you can revisit the recording. So let's see, but there's been so many good questions that it's been a bigger investment of our time together, but that's fine with me. I love questions. So let me go into this Mentimeter poll. Okay, so for those of you that joined late, again, you can join the poll here at this URL, or go to menti.com. You can do it on your phone or your web browser. Enter this code, and for those of you that were already in the poll, you should be able to just open up your session and continue. But let's start with one question here, right? So let's see if we can apply what we've learned, right? A lab mate comes to you and says that they need to adjust a CD69 positive gate and a CD38 positive gate across different CD4 T cell subsets in the same sample. What would be most helpful for this, right? What would be your advice to your lab mate? And it looks like we've got almost unanimous decision for the second choice, which is to use clone gates, right? So clone the CD69 gate and the CD38 gate across the CD4 subsets. And I think I see, let me check the chat again. Okay. Let's see. Some of these questions I think I'll come back to at the end of the demo. Let's see. Okay, somebody was asking about color gating in FCS Express. I'm not sure what you mean by color gating. I guess color coding populations in the same plot, like like the way you would do in Diva. I think I understand. Yeah, I mean that's an interesting feature request. Something we can think about, as we're developing the next version of FlowJo. I think it's just, it's not that we don't think it's a cool feature, but we invest time into other computational tools rather than visual, changes to the plot. So, I totally hear where you're coming from. But yeah, it usually just comes down to priority, really. But that is a cool feature. I do like that Diva does that you can, change colors in the same plot. Okay, there was another question. Oh, there was a question about, can you do the opposite of merge gates and create two separate bisector gates from quadrant gates? That's a good question. I want to say no, but now I want to go back to the UI and just double check. Create merge quad gate, create. Yeah, I'm thinking no, Elena, but that is a good question. And maybe it's possible, and I'm just unaware of how. Now, what you could do is you could do the auto merge, and that'll give you automatically a bisector and a quad, but that's really just if you haven't drawn any gates at all. It sounds like what you're saying is if you had the quadrant gate, could you just get the bisector gates? Um, so that's a cool feature. I'll have to think on that. Okay, and then let me go back to the poll. We're running a little bit of short time. Okay, so pretty unanimous decision, right? I think we're all got that one pretty good, which is basically, yes, if you use clone gates, that would be really helpful here. Okay, let's do another question, and I'll check the chat while you all are working on this one. Now, let's say I have this plot here, right? In my analysis, I did monocyte-derived dendritic cells, I did plasmacytoid dendritic cells, and there's some stuff that's in that same plot that I don't want, right? So it's not as simple as just going up to the parent and grabbing the parent population if I want to look at these together, right? So let's say I've changed my workflow and now I need to look at these populations together. But again, it's not going to be as simple as just going up to the parent because I don't want this stuff here. So how can I get these two populations? So I'll let you guys answer that for a second. Let me go back to looks like there's something in the Q&A. Okay, looks like somebody has a question about quantifying a stain with green and minor chakras deep red. Mariska, that's a good question. It's probably going to be a little application specific. If you want to send me an email, we can try to tackle that by email. But in short, we probably have some tools that can help you with your analysis. We don't have any kind of like modeling. If that's what you're asking. If you're looking for some kind of like what we see for proliferation dyes or cell cycle dyes or kinetics, like we're not going to have any kind of a model that's specific to what you're asking, I think. But if if you're just looking for advice on, workflow and how best to go forward with your analysis, I'll probably just need a little bit more information, and then I can definitely help with that. Yes, and please email me. I myself am not a parasitologist, so anything more you can share, I'll be able to give a better answer. All right, let's see how we're doing. So it seems like most people are saying the AND gate, right? So you say MDC and PDC. We got a couple people saying a NOT gate, an or combinatorial. All right, are we ready for the answer? I think so. So this one's tricky, right? And it would be helpful maybe if I showed you the little icons for the Boolean, right? But the answer is actually going to be the OR gate, right? Because an AND gate is going to give me only cells that are double positive for CD123 and CD11C, right? So because here I'm giving these two populations and it has to, with the AND logic, you're saying it has to be this and that, which would capture anything double positive, right? These are mutually exclusive populations. The NOT gate is going to give me everything at the bottom, right? So if I select these populations and say not, it's going to be not MDC, not PDC, and all that leaves is this, which I'm not interested in. The or means I want a cell, whether it's this or that, and that's going to give me both, right? So let's go back to that icon and I think that'll help us here. So here, let's look at this picture again, right? The AND gate will only take a cell that meets the criteria of both populations. The NOT gate takes what is outside the gate. The OR means it's everything, right? So that's how you merge populations when you want them both. Okay. The combination gate, I guess, could work, but you get a bunch of populations you don't need, right? And all I needed was these two together. Okay, so a little tricky. I knew that one would be a little hard, but I wanted to see if we could talk through it together. And the good news is once you do the Boolean function in FlowJo, you'd immediately realize that you may not have gotten what you wanted, right? So you can always just redo the Boolean. Okay, so let's do one last question here, and I'll check the chat one more time. But which tools do not directly handle sample-to-sample variability? Okay, so if I have variability in gate placement between samples, right, which of these tools is not going to be helpful for that? Okay, so think through that for a second. Let me check the Q&A. Okay. Oh, I think I answered these already. Great. Good one, those. Perfect. Very nice. Okay. Looks like we're almost unanimous again. Not 100%, but we're close. Okay. All right. Give people one more second, and then we'll go ahead and show the answers. And again, looks like somebody raised their hand, but just a reminder, I cannot unmute participants. So if you have a question, you'll have to put it in the chat or the Q&A box. All right, so we're right at the hour. So let's go ahead and wrap with the quiz here. So correct answers are, enter key here, there we go. Clone gates and workspace templates, right? Remember, clone gates are only within a sample, right? So it's going to do within population variability, but not sample to sample variability. Okay. Workspace templates, super powerful, right? They give us a way to copy gates, copy an entire hierarchy onto samples in the same group, but that's just going to do the placement, right? It's not going to necessarily address sample to sample variability. So you'll still have to come in and change the gates through various mechanisms, which could include Elastigate, right, for gate adjustment. And then EasyDaffy, it actually does handle sample to sample variability, because once we do the training, which again, we can be very selective on how we do the training. Once we do the training for it to find the populations, it should be able to find it in a sample specific manner in different samples. Right? It's a little bit more a different approach than Elastigate, which is just using kind of visual information to move gates. Right? EasyDaffy is first kind of defining those populations in the high dimensional space, and then using those definitions to find populations in subsequent samples. Okay? But it should still do a better job than, say, something of just copying and pasting a gate without adjusting it at all. Okay. So nice work, everybody. Okay, so now I'm going to go into the software and let's see if we can quickly at least demo some of these, just so you have a sense of where they are. But all of this information is well documented on our website, so you can always revisit the information. Okay, let's talk about clone gates first. Okay, so here's my FlowJo workspace. So let's say in my CD4 hierarchy, I've got this interferon gamma population. And I've got the same interferon gamma gate on my CD8 T cells, right? So here's CD4, here's CD8, but I have the same interferon gamma population that I'm trying to identify, okay? So if I had a lot of other subpopulations for interferon gamma, if I need to make an adjustment, right, on one gate, let me go ahead and open that so we can look at it. If I want to make an adjustment on one gate, I'll have to do that for individually, right, for each of the different subsets, unless I'm using something like Elastigate. But even Elastigate won't put it in the exact same coordinates, right? It's just going to do what it thinks is best for that sample. So if I want to sync it at least within the same sample, all I have to do is, and actually let me open up the CD8 too so we can see it. There we go. Okay. So here I've got the interferon gamma gate, but one's on CD4 cells, one's on CD8. I'm going to select the two gates that I want to clone within the same sample, right click and say clone gates. Right now, notice how I have now this little kind of 3 green diamond icon in front of the gate, and that tells me it's cloned. So now if I move a gate in the CD4, the CD8 will follow. Okay? If I do the reverse, if I change the gate in the CD8, the CD4 will follow, right? They are now basically one and the same. And when I make changes to one, the other will also change. So that's quick and dirty clone gates there. Okay. If you want to undo it, right, you can just select the populations, right click, see how it's reminding me the clone gates are on. I'll just click that and turn them off. All right, so that's clone gates. Let's see, what do we do next? Let's talk about maybe Booleans, right? So let's say I have these two populations that I've gated for. But now I want to see them in different combinations, right? And I don't want to go back and redo quadrant gates. I want to just leverage the information I already have here. So I want to now see cell types that are different combinations of interferon gamma or phospho-ERK. So I'll select those two populations. In the FlowJo workspace, I will navigate at the top of the ribbon to the workspace tab, or sorry, the tools tab. In the tools tab, there's a Boolean section. Now, if you ever can't see anything in your activity ribbon, go to this ribbon manager here and make sure you drag it in. So here's the Boolean functions. You can drag it in, quads, drag it in. So sometimes they disappear because we accidentally drag them off or something like that. So if they're ever missing, don't panic, just bring them back from the ribbon. Okay? So if I have these two populations, notice how when I only select one, the only thing I can do is a NOT gate, right? It won't let me do anything else because I don't have another parameter selected. So now that I've selected both, it's kind of unlocking those other choices, right? So I could do an OR gate, which would basically merge those populations, an AND, which would only give me double positive populations, or a NOT, which would give me everything that's negative for these markers, basically, right? But if I want all of those combinations, I can say create combination gate, right? It's just going to ask me what do I want to call them, say okay, right? And there's all the different combos. I've got the NOT gates, double positive, single positive interferon gamma, single positive phospho-ERK, and double negative. Okay, so almost like I had a quadrant gate. But keep in mind, again, I'm going to double click that, let's say the double positive population. And if I go up a level, I'm only brought back to the single positive gate that derived the Boolean, right? There's no quadrant gates here. Okay. So if you decide, oh, you know, I feel like I want the double positives to include a little more interferon gamma, right? You'll have to go back to these gates and do the adjustments. So the Boolean populations will be live, but they themselves don't have gates, right? They're derived from the single gates. Okay. Now let me check, looks like some more stuff came through in the Q&A in the chat. So, can you clone quad gates? Oh, that's a good question. I don't know if I've ever tried to do that, but why not? Let's try it. So let's see, if I do, I'm gonna pick something else. Let's do HLADR and let's do CD38, why not? Let's make a quad gate. There it is. And then if I have those quad gates and I put them, so right now I have them on CD3, but let's say I put them on CD8. And if I did pick these, let's see what would happen. Well, now that I think about it, it's probably not going to be possible, but let's see. Well, it says clone gates. So if I do that, and then I'm going to open that same hierarchy here, HLADR and CD38. Now I'm just testing this because I don't know the answer to the question, but let's see if we can figure it out. So if I move this, does it move? It looks like it did. So then yes, you can clone quad gates. So I went ahead and multi-selected all of them, which you probably have to because they're not necessarily individual rectangle gates. But yeah, it looks like I'm doing it now and it is synced. So yes, you can clone quad gates. I'm gonna go ahead and delete these just because I don't want to confuse myself. That's a good question. All right, so let's see. Let me go back to the Q&A, see if I missed anything there. Answered that one. Great. Right. Oh, another question was, can you apply this to the group? Yes, I believe you can. I believe if you apply clone gates to the group, the clone gates still sync within each sample, I believe. Okay. All right, so we talked about, let's see, we did clone gates, we did Boolean. The other shortcut I actually learned the other day myself was that I showed you the NOT gate, which again, in this case, if I had the singlets, for instance, and I picked that gate and made it a NOT gate, it would get everything outside the singlets, right? Maybe this is a bad example. Let's do lymphocytes. Right, so we talked about that in the presentation. If you just want to quickly see a NOT Boolean, if you go into the active gate dropdown in the graph window and select the gate, And see how it says active gate events inside, right? That means you're displaying the events inside the gate. If you turn that off, now you're displaying the events outside the gate. And see how that percentage went down to 11. And if you go back to your hierarchy, look at the lymphocytes, right? It has a dash through the population, meaning it's a not gate. Okay, turn it back off. Now the percentage is what's inside the gate. So that's just a quick shortcut. But if you do the Boolean not, it's the same idea. Okay. Now, so what's the alternative, right, to Booleans? That would be something like the merged quads. Okay, but for that, you're going to need to do the bisector gate. So let's pick maybe, for instance, CD8 T cells. I'm going to pick that population, open it up, and let's maybe do a bisector gate on, I already have HLA-DR open, so let's go ahead and do that. So bisector gate, right, it's only available in a histogram view. That's this, when you hover over the range gate tool, you'll see bisector tool. I'm going to split it here. We'll say this is HLA-DR, right, positive and negative there. Okay, so there's the CD8 population. There's the positive and the negative bisector. Let's pick another marker. Let's say perforin. Okay, I'll do the same thing here. Bisector gate, perforin, negative and positive. Yeah, so let's say I've done all this work. I have all of these existing bisector gates, but now I want to see combinations. Now, if I want a specific combination, like I only want perforin double positive and HLA-DR double positive cells, I could just do a Boolean, right? I don't have to do a merged quad. So if you want something specific, you could just do a Boolean for it. Okay, but if I do want the merged quad, I have to pick all 4 bisector gates, right? The negative positive for one parameter, the negative positive for the second. In the FlowJo ribbon, same section as Boolean tools, there's a quad section. Here's my options, right? I can do a merged quad or a merged curly quad. If I only want a gate, maybe not a Boolean, but I still want a gate, I want double positive HLA-DR perforin. Actually, let me go ahead and pick these again. There we go. Or you could also do like a merged rectangle gate as well. So let's do the merged quad gate. Okay. Right, and there it is. But this time, this is different from the Boolean because I actually have a quadrant gate representation now, right? So here's the HLA-DR, here's the perforin, there's the 4 quadrants. Okay? And again, you can still make adjustments, but now I can see that distribution in a single plot instead of having the different histogram views that you had before, right? Instead of having to leverage these, right? I can leverage something like this. All right, so I think that was all in terms of the advanced gating, for existing gates. So let me go ahead now and I'll quickly try to do Elastigate and Easy Daffy as well. But first, let me check. It looks like something came through in the chat. Is the merged rectangle gate made from 2 range gates? Yeah, I believe so. So let me see if I can, for instance, here, I've got this one. There it is. Okay, so here's a good example. So here I have the CD4 T cell population, and I've got the interferon gamma gate, right? Not bisector, but I think just a single range gate. Here's the phospho-ERK. I'll go up one there. Okay, so these were standalone range gates, right? Not bisector. So there's the 2. And then, so if I select those 2 populations and say create merged rectangle, right? And I'll just leave it as rectangle gate. Let's go up a level. Right, here's what it gave me. Okay, so there's the rectangle gate off interferon gamma and phospho-ERK. Q&A's got some stuff. Okay, let's see. Oh, yes, somebody asked about the cache enable. Yes, good call out. So we were talking about Boolean gates and how if you have a workspace full of these, it's going to start to increase the the computational burden of the calculations. We can help that along by caching Boolean. So in your preferences, which is this heart-shaped symbol at the top right corner, hit that. It's going to open up your preferences window. Now let's see if I can remember where it is. Is it in gates? Nope. So let me go back. Maybe it's in graphs. One of those things where I just end up clicking around until I remember where it is. Ah, there it is. Okay. So in your preferences, it is under performance, which makes sense now in hindsight. So performance, there at the very bottom, there's this box that says cache Boolean gates. And so basically that'll retain the gates in the memory and just kind of locally while you're working. And that way, as you're making calculation changes, like moving gates around and things like that, it won't be so laggy to recalculate the stats. Okay? So that should be helpful if you enable that. Okay. Thank you for reminding me to do that. There was another question in the Q&A that says, can you use the active gate function to not to population at the same time? Ah, that's a good question. I guess we could try it. Let's see. So if I go into lymphocytes as an example, and let's say I draw another gate here. Yeah, that's fine. Okay, so will it let me pick both? It might not. Active gate. Yeah, it looks like it only lets you pick one. I guess that's why it says active gate instead of active gates. So I don't think you can multi-select and do the active gate shortcut. But if all you want is everything that's not in it, then you could do the Boolean, right? You would just select those two populations and then do the not Boolean. But the shortcut, I suppose, will only work for one gate at a time. That's a good question. Okay, let's see. I might have missed something else in the Q&A. No, that's the one I just answered. Great. Okay. All right, so let's see if in the next couple minutes I can show you Elastigate and EasyDaffy. EasyDaffy, I may just kind of show you where the buttons are and show you a completed workspace because it does take a couple minutes to calculate and we're a little over time already, so I don't want to keep people too long. So I'm going to close this workspace because I've been doing a lot of changes here. So I want to open something fresh. Get something here. Okay. So Elastigate is a plugin. It's available in the FlowJo Exchange. Okay, it's at no additional cost or anything like that. It's just like an application you have to install so that it becomes available in your plugins section in FlowJo. Okay, so if you have questions about how to set up plugins, you can look at our documentation on our website. It covers it very nicely, or send me an email and I can help walk you through how to get that installed. Okay. So once you have that installed and set up, right? Let's say I've got this hierarchy here. I carefully gated, singlets, lymphocytes, dead cells, all of these cell types. And I'm confident, at least in one of the samples, that I've placed everything nicely. So it's going to be a nice training sample to run Elastigate on. So what you'll do is, and I applied that gate to the rest of the samples, but remember when we apply gates, it just copies the exact coordinates. It's not doing sample to sample variability. So how do I account for that? Well, I could use something like Elastigate. Okay, so I'm going to select the sample, go to Plugins, Elastigate. Okay, a couple simple questions. First question, what's the training sample, right? What is the algorithm learning from? Okay, I did all my gating on the first sample, so I'll do that one, but you can have more than one, which might help the algorithm perform better, honestly. Which ones do you want to apply it to? Okay, I'll uncheck the one that I used for the training. Which parts of the hierarchy do you want to apply, right? Maybe you don't want Elastigate to do the full hierarchy. Maybe you only want it to do some of the gates. Okay, so uncheck what you don't want it to do. I'll have it do everything. Now, a couple of toggles down here. Most things I don't change. The only thing I would say that you might be interested in using is this box down here that says ignore non-matching populations, right? So basically that's addressing the caveat we talked about in the presentation, which is that if populations are missing because the biology is different, like you have a stimulated versus unstimulated sample, you have an FMO versus a full stain, something like that, you're basically telling the algorithm, if you don't see something in the general vicinity of where it should be, it's not there. So I don't want you to gate, right, the next biggest population over. Okay, and that's just to try to help the tool along. Okay, now another workaround would be you train it on a sample that matches the biology, right? Only train a stim to apply to stim samples, things like that. Maybe don't use FMOs or skip the missing population in the FMO. Okay, there's other workarounds we can do, but that's at least just one attempt to address that. Okay. So after that's all done, I'll say start, right? Notice how my gates are moving back there, right? So Elastigate's already learning, adjusting gates and applying them to the rest of my samples. Okay, so it's fairly quick. Might take longer if you have more samples and a longer hierarchy. Okay, but you can see it's chugging along here. And once it's done, The only evidence you'll have it even ran is there'll be a little star that says Elastigate, but otherwise all your gates are going to look like just user-defined gates, right? They'll still have the vertices where you can make adjustments. Okay, so this finished running, so I'm going to close this now. Right, there's the little Elastigate star, right? Just letting me know that it did complete. And let's, open this up and see how it did. Right, so if I open up all these gates, right, you still get the gates for the adjustment. Okay, so if I don't like anything that it did, right, I can come in and still change those around, right? Oh, I want this to move over a little bit. There, I can do that. Okay, if I go to the next sample, right, I think it actually, see how that CD4 population moved? Right, Elastigate caught it. There's the next sample, right? It moved to the left and it gated it very nicely, right? So just gives you a sense of what it's trying to do for you, okay? So again, this tool gets covered a little bit more detailed in that supervised webinar, so I'd recommend you watch that if you want more detail. So I'll quickly show you Easy Daffy. Let me close all graph windows and let me close this workspace as well. Right, and I'm going to show you one where it's already complete since we're kind of short on time. All right, so here's if we want to run Easy Daffy. Now let's say, for instance, right, this is all the same data set I've been using over and over in these workspaces, just approaching it differently. So let's say I've gotten down to my analysis and I have this CTL population, which I've defined as just being double positive for interferon gamma and perforin. And I'm really interested in that population and finding not only that population, but maybe populations that look like that population. And so I don't mind not having gates as long as this algorithm can find me cells that look like that. Okay. So what you would do is, in this case, right, I recommend you take your training sample and move it into a separate group. And that way, once you have your Easy Daffy node, you can drag it to the rest of the group. Okay, and we go through all this in the Easy Daffy webinar. But essentially, I'll say, okay, I want Easy Daffy to find, I'm mainly interested in the CTL, but the more parent populations you offer, the better job it might do because it has more opportunity to learn of what's the target and what's not the target. So if CTL is the target, maybe I want to at least feed in CD8, but might be helpful to feed in CD3 so that you can also learn what's not a CD8, right? Throw the CD4s in there. So you'll pick the CD3 population, workspace plugins. Easy Daffy is also a plugin that needs to be installed along with the R program. Select Easy Daffy. Okay, I generally leave all the defaults. I don't typically add anything. You can overlay the population. That just creates a visual in the layout editor where it'll compare the Easy Daffy population to your manually defined one, if you want that. There was a question earlier about, well, how many parameters do you need for the calculation? So again, if you leave it unchecked, which we recommend, it's basically Easy Daffy will only pick the parameters that it thinks are most important. If you want it to use all of them, you can say use naive Daffy algorithm, but it may not be as accurate and it's going to take longer to run. Okay, so I would say as a first pass, you're probably going to want to leave it off. Last box doesn't apply to you unless you're doing RNA-seq data. Okay, so you'll say okay, but again, I don't want to let this run because it's going to take a few minutes. But once it's done, here's what you'll get, right? You'll get your Easy Daffy node just to tell you it's complete. And here's my populations down here. I've got the Easy Daffy CTL population, the CD8 T cell, CD4. I just realized these all say 0 because I may have accidentally deleted the calculations. But you should have percentages here. And so those populations are basically the populations that Easy Daffy tried to find using the algorithm there. Okay. So this is all nicely outlined in that webinar. I think something happened with this workspace when I ran it before, but maybe while I go back to our summary slide, I'll close this and then maybe I'll let a new one run in the background. And that way we're not wasting time, we can move on, but you can see what a completed one looks like. And I'll tackle any last minute questions. So I'll go back here. Maybe I'll run it on the CD8 just so it's faster. And we'll say Easy Daffy. I'll do the overlay. Maybe that'll come through. Okay. I'll say okay. Maybe I'll let that run and see if we've got any last minute questions here. Okay. Chat. And I think there were some questions earlier that I didn't get to. So let me see if I can get back to those. Questions. Okay, so somebody said, what is the min? Okay, this was one that I wanted to wait till we got to the easy DAFI. Someone said, if it's not clustering on the basis of markers that I did not include, then on what the classification of the cells are dependent on. So that's a good question. So I think this graphic will help explain that well. So this ran faster than I thought it would. So here's the CTL population I asked Easy DAFI to find. Okay, so there's the CTL I manually defined and the Easy DAFI population. Here's my CTL gate, which is in black. The burgundy is the Easy DAFI population. And you can see it almost perfectly fits in the gate, but there's some extra red dots that weren't in my gate. So where did those come from? Those came from the fact that Easy DAFI identified cells that were interferon gamma positive, were perforin positive, CD8 positive, but they were also maybe had some CD38, which I didn't use CD38 in my upstream hierarchy. Now, if it's important to me that these cells not be CD38 positive or that they be CD38 positive, then I'll include it in my hierarchy. And that's more training, right? More guidance, more supervision for the algorithm. But if this is at minimum what I want, and I want it to consider other markers, it will consider those. And it's showing you the ones that it found to be relevant for finding this population. So basically, Easy Daffy is telling me, if you're interested in just perforin and interferon gamma double positive CD8 T cells, just know that CD38 plays a role, right? You can also identify those cells if you consider CD38 expression, right? So it's just considering other markers that help define that cell type that you gave it. Is the short answer. Let me see if I missed anything else. See, scroll down. Oh, there was a question at the bottom. Does the scaling transformation matter when running Easy DAFI? Oh, that's a good question, Thomas. I would say for any algorithm where you're leveraging what we call graph space, so the visual separation between populations or events, transformation is very important. For Elastigate, probably not. I think if you have good separation, I think that's still going to be helpful. But for Easy DAFI, since we're not supervising necessarily like the populations that get retrieved, I mean, we are with the gate, but ultimately the clustering that initiates the tool is unsupervised. I think a good transformation where you've got a good separation between events is definitely going to be helpful. So for those of you less familiar with high dimensional workflows, right? If we have events that are very smashed together, algorithms have trouble identifying them as separate, right? Because we're not using the MFI values, we're just using how far apart are two events on a plot is the simple answer. So the more distance you can create between what you think to be different populations, the algorithm has a better chance of also defining those as different. Whereas if they're very smashed together and you got a lot of white space on either end, there's less separating those populations, so they might be considered the same. Okay, so if you think things need to be separated more, do so. Yeah, definitely a good call out. Okay, so let's see. I think I answered everything in the chat, but a lot of questions came through at the same time. So if I missed a question from you, please don't hesitate to reach out by email. Maybe as we wrap up, I'll do one final poll question here, which is I just want to know what people are excited about, right? We covered a lot today. I feel like most questions came around clone gates, which I thought was kind of funny. So, everything we've learned today, clone gates, Booleans, Elastigate, merged quads, Easy DAFI. I'm just curious, what are people excited to apply to their data? So if you still have this poll open, I'd love to hear your thoughts on what you found to be exciting. Everything. I feel like every time I do a word cloud prompt, I always get at least one everything answer, which is great. And let me see if I missed any other questions while I let people answer this. Yes, Prabhat, I will put up my email once this poll kind of finalizes. I'm going to put up my email so everybody has it one last time. So I'll say that I answered that. And then somebody asked, when analyzing a 30+ panel of spectral data, I sometimes have to adjust the axis. Will the modified axis be considered by the plugins? Yes, that's a good question. So gates are kind of like I guess I'll take that back. So if you do change the transformation, the coordinates of a manual gate might not adjust appropriately. So if you do changes to your transformation, you may have to go back and look at your gates. So you might want to get all your transformation worked out before you establish gates. Now, if we're talking about something like Easy Daffy, you'll want to establish your transformation before you do the Easy Daffy clustering, because the Easy Daffy clusters are frozen. So if you go back and make gate adjustments to the populations you used to find the Easy Daffy clusters, that's not going to change the subsequent Easy Daffy clusters. Okay, so you'll need to rerun the tool. So I would say all in all, it's usually good to try to establish your transformation before you place the gate on the plot. Because otherwise, if you start making all these transformation changes after you're gating, you might have to be doing some gate adjustments. Or you could run Elastigate again. All right, so let's see. Looks like a lot of interest around clone gates. Easy to feel like clone gates are kind of winning out here, which is exciting. All right. Well, I'll leave that open for just another second, but it looks like the answers are kind of tapering off there, which is fine. So I'm going to go ahead and just for anybody that needs to jump off, I'm going to go ahead and put my email here just one last time. So, and I'll also thank you all for your attention. I really enjoyed today's webinar, a lot of active questions and discussion. I hope you found it useful for your studies, for your analyses. And of course, as promised, if you run into any issues while doing your analysis or trying out some of these tools you learned about, there's my email. Don't hesitate to reach out. I'm also available through a quick LinkedIn search for those of you that use LinkedIn. You can reach out through that avenue as well. And the other thing I'll remind as well is that we have a lot of learning resources on our website. So don't take those for granted. In the learning tab of flowjo.com, we've got webinars, including this one, which will be recorded. There's the supervised auto-gating one if you really want to learn more about the Easy Daffy and Elastigate. FlowJo University, short tutorials, little videos to walk you through tools. Documentation, which is my favorite because it usually has screenshots and paragraphs and really just kind of digest that information. And then tech support. If you're installing these plugins and they're failing on you, they don't run, they don't give you the outputs, reach out to me, of course, happy to help, but the direct line will be flowjo@bd.com. Very responsive group of application specialists that can help with plugin install. All right, so let me double check the chat. A lot of thank yous. Thank you all. This was very fun for me as well. So I'm going to kind of go back to that poll just to see if there are any parting comments there. But I think I'm going to go ahead and stop the recording here in a second, but I'll hang on if there's any remaining questions. And I will wish you all a wonderful rest of your day for whatever's left of it, depending what part of the world you're in. Otherwise, also have a great weekend, and thank you for joining today.